My name is Nicole Seiberlich, and I am an associate professor of radiology at the University of Michigan. And I am going to help us switch gears to the discussion of medical imaging. So I have the very great honor of introducing Dr. Dan Sodickson today as our speaker now on the medical imaging side. And to those of you who are in medical imaging, Dr. Sodickson needs no introduction. He's really one of the superstars in our field who is known by everyone. But for the rest of the audience, I am going to offer a brief overview of his training and accomplishments so you can know him a little bit better. Dr. Sodickson is the Vice Chair for Research in the Department of Radiology at NYU Langone Health, the Professor of Radiology and Physiology and Neuroscience at NYU Grossman School of Medicine, and Professor of Biomedical Engineering at NYU Tandon School of Engineering. And in addition, he leads NYU's Center for Advanced Imaging Innovation and Research. He graduated from Yale with degrees in physics and humanities before going on to pursue an MD at Harvard Medical School and a PhD in MIT. During this time, he began his work in rapid magnetic resonance imaging in the form of parallel imaging and founded this field of work, which is key to clinical MR today. We're using this in almost all the scans that we do. In addition to the titles that I already explained, he holds a slew of additional titles, including the director of the Bernard and Irene Schwartz Center for Biomedical Imaging, and he's the co-director of a new institute at NYU, Tech for Health, designed to bring emerging technologies such as continuous sensing and artificial intelligence to biomedicine. So we invited him to give this lecture because of his depth of understanding of not only MRI, but also other imaging modalities like CT and PET. And he's also an expert in the process of translating innovations in medical imaging to clinical practice. And so we really couldn't think of a better person to be giving this lecture today. But Dr. Sodickson is an excellent educator and a multidisciplinary collaborator, which is what we were going for in this series. But he's also one of the unique minds in medical imaging, and he'll offer us an inspired view of the current status and future prospects of this field. So without further ado, I'm going to hand the stage over to Dr. Dan Sodickson.
Thank you so much, Nicole. I will do my best to come anywhere close to that introduction. All right, well, I am particularly delighted to be speaking in this cross-cutting seminar series. And I'm also excited to follow Urvashi, who has broadened my horizons already in the talks that she's referenced, where we were coordinating our lectures. My job today is to provide the complement to her introduction, namely an introduction to medical imaging for radio astronomers. And since this is an introduction, I'd like to start at the beginning. In the beginning, there was darkness. And then there was light. From the earliest dawn of human history, people have sought to extend their vision. They built tools to help them see further and see clearer. And these were the origins of astronomy. But what of my field of medical imaging? I would put it to you that medical imagers are the explorers of inner space. And so whereas the tools of astronomy look outward into the cosmos, our tools look inward to see what we ourselves are made of. And as you've already heard from the first talk in this seminar series on through Urvashi's talk, there are many points of connection between these fields, despite the very large differences in perspective. There are also particularly strong connections, as you've now heard in some detail, between radio astronomy and MRI, despite the vastly different spatial scales and the notably different equipment that we use to make our measurements. And in particular, these fields share the same area of the electromagnetic spectrum, the same structure for our signal space, and also the same form of our inverse problem. And I'll try to dig into each of these in the course of the talk now. So here's the overall outline of my journey with you today. And as Urvashi mentioned, we chose a kind of common overall structure. So I'll begin by giving you some basic history of medical imaging and try to outline some of the key operating principles of the 4 cross-sectional medical imaging modalities that are most frequently used today: CT, PET, MRI, and ultrasound. Then, though, I'll try to really focus in on MRI and present it from a perspective that I hope will be at least somewhat familiar to the radio astronomer. And then in the end, I'll pull back out to talk a little bit about the future of medical imaging. First, my disclosures for your consideration. Okay, history. So in the course of this highly abbreviated history, you're going to notice some ongoing themes about the progressive development of enabling technologies, and also the development of increasingly abstract transforms that allow us to convert the measurements from those devices into images. Now, the first imaging technology was not one we invented. Biology did that for us. So it turns out that in the early oceans, it was a significant competitive advantage to be able to tell here from there. And so a veritable arms race developed to form biological devices like these, this trilobite compound eye in the Cambrian era during the Cambrian explosion. From there, it's just a hop, skip, and a jump, really, to the human camera eye, which incorporates neat bells and whistles like lenses for focusing light. Now, once we had eyes and brains of our own, it took us a little while to emulate and ultimately improve upon nature. And I would argue that the next big step came when we learned to bend light with lenses. Because of course, as you in astronomy all know, bending light is what gives us the gift of magnification and brings the cosmos a little nearer to our grasp. Of course, also, bent light gave us our first imaging tubes. Now, that's outer space. In order to have access to inner space, we first needed to be able to see behind the surface of things, see through the skin. And fortunately, in 1895, Wilhelm Conrad Röntgen discovered just the thing in the form of X-rays. And X-rays, of course, can blast their way right through previously opaque things, being attenuated progressively by any dense structures in their path. So that's a great probe, but along with this probe came the next very important transform, which is that if you think about the shadow pictures that result from an X-ray, they're actually sums of 2D cross-sections of the object density all across the path, along the path of the X-ray beam. And so it was then those shadow pictures, those projections, which gave us the field of radiology. Now, the next trick was to try now to untangle those individual cross-sections out from underneath those sums. And it turns out a good way to do that was to gather projections from a lot of different angles, or multiple different complementary projections anyway, and use mathematics to try to sort out what went where. This is called tomography, and it got its start in the magic decade of the 1970s, or at least came into its own then. Why the 70s? Because that's when the computers that had sufficient power to do this mathematics came around. It's also when people were able to start building the big tubes we associate with modalities like CT, PET, and MRI. So how do we do this sorting out? How do we undo that summation transform? Well, here's a sort of intuitive algorithm. There are many algorithms now out there, and I'll get to the inverse problems that Urvashi just discussed in a little bit. But just to give you an intuitive sense first, here's a way. So let's say you have a projection going through this schematic object in this direction. Well, what you can do is take that projection and smear the intensities back along the direction the projection came. And so you see with this example, you get these two smears of brightness associated with each of the dense objects in this structure. That makes sense because as of yet, we don't know where these objects are along the path of the beam. But now you do the same thing from another angle, and another, and another, and sure enough, you start to see the brightness accumulating in the regions of the object and spreading out elsewhere. And in fact, if you take a complete set of projections and perform this procedure, you get a relatively faithful copy of that image cross-section. You may see a little bit of blurring here from the residual smearing. That's something you can take care of with a little filtration, and you're good. So when you do this procedure with X-rays, it's called X-ray computed tomography, or CT. But it turns out there are many ways to create projections, not just with X-rays. You can create projections with a wide range of probes across the electromagnetic spectrum and elsewhere. This is the analog of the many different sort of flavors of astronomy in different parts of the spectrum. So for example, you can create projections with positrons, which gives us positron emission tomography, or PET. Basically, PET, if you will, is CT turned inside out. So instead of sending X-rays through a body, you actually inject radio tracers into the body, which accumulate, and then these radio tracers decay, emitting positrons. Those positrons encounter very quickly electrons in the body, and annihilate, creating oppositely directed gamma rays, which aren't really as much attenuated as X-rays, but they blast out in either direction. So what you can do then, and by the way, this is then up at the top of the electromagnetic spectrum. What you can do then is array detectors around the body. And then whenever there's an annihilation event, you can capture the two coincident gamma rays in appropriate detectors, and you know that that event occurred somewhere along the line between the detectors. So over time, you build up a bunch of events, and then you can just group them into similar angles. And lo and behold, when you do that, what have you got? A projection. And so you can collect your different projections now and reconstruct it just the way you would have CT. Okay, so that's the top of the spectrum. But we can also produce projections down at the low end of the spectrum in the radio frequencies. And it turns out that in magnetic resonance, if you put on a strong magnetic field, it'll align nuclear spins, for example. And when you knock those spins out of alignment, they will emit radio frequency fields that are detectable. So that's great. But at this point, you have no way of sorting out what's going on where. All the spins are behaving the same in the sample. So it turns out there's a very simple trick to solve that problem. You just vary the field across the object. You make it weaker on one side and stronger on the other. And then the RF that's coming out from one side of the sample is at a higher frequency and on the other a lower, which means if you take a frequency analysis for a transform of your signal, what do you get? A projection. And we're back. And then the last one I'll show is that you can, of course, create projections with sound as well. And we were not the first species to do that. Other species like bats figured out echolocation long ago. And you know that from the return time of the echo of a ping that the bat sends out, it can tell how far its tasty prey, like this moth over here might be. And you can, if you can focus your beam, create lots of different lines and generate a set of images from that. But what if you don't have perfect focusing, like the bat doesn't? Well, it turns out you can still use projections. And ultrasound basically uses a higher frequency of sound. You have a transducer here, it sends out a pulse. It can pass through tissue, but it bounces off some of the interfaces in density in the tissue. And eventually that pulse comes back and is detected and so on. And if you now were to look at the signal as a function of return time, what do you get? A projection. So this notion of reconstruction from projections really connects all of the medical imaging modalities, the cross-sectional modalities that are in common use. And here's just a summary of those modalities I just talked about. Many of them, all except ultrasound, are still stuck in these big tubes. Ultrasound has the nice handheld transducer. There are different native contrasts of the different types of imaging, and that's the analog again of the different types of images in astronomy. And there are different orientations you can take the images in. The probes have associated with them different safety considerations, like CT and PET have ionizing radiation, ultrasound and MRI do not, and so on. But at this point, you may be wondering, Dan, what's the connection between all of these projections and radio astronomy, as I just saw with Urvashi? So let me now dig in a little bit and take the covers off the MRI machine and try to show you, at the beginning anyway, how MRI hardware works in 3 simple steps, or to be more particular, 3 electromagnetic fields, which we use to generate, detect, and localize our signal. So the first of these fields is our so-called B0 field. It is our constant magnetic field that we put on. It sets our signal strength. How? Well, the difference in Zeeman energy between different spin states, say up and down for this spin a half, is proportional to the magnetic field strength. And so there is therefore a thermal population difference. And that population difference gives us what we call our spin polarization. Now, it's important to distinguish this from light polarization that you might use in astronomy. In this case, we mean essentially the population difference between up and down. And that polarization then sets the level of our magnetization, how much the spins are aligned in one direction or another. That scales as the square of the magnetic field strength. As you already heard, the field strength also sets the operating frequency, the so-called Larmor frequency at which these spins precess around the magnetic field. And it also defines the form factor of the usual MRI machine. which is this big cylindrical electromagnet, which is nice to get strong and homogeneous fields. Okay, that's field one. The second field is our so-called B1 field, the radio frequency field. We use this first to excite our signal. So we use it to tip the spins off of the vertical and set them precessing at the Larmor frequency. How do we do this? Well, it turns out that a simple tuned loop will do the trick to create oscillating radio frequency fields. Or we could use fancier structures like this bird cage arrangement surrounding the body. And it also turns out that the same structures can be used to detect signal, because the changing flux from the magnetic field that this little spin dipole drags with it as it's precessing around can be detected in this coil. So that means that our signal then has an amplitude, it has a frequency, but we also do phase-sensitive quadrature detection in order to get a phase as well, which is where along this cycle the spin is. And you'll see that that makes an important difference later on. Okay, well, those same detectors can, of course, detect noise as well, which may come in the form of Johnson noise in the electronics, or it may be random currents in the conductive body that happen to lie within the bandwidth of our detection. Now, I should mention that the radio frequency wavelength that we typically use at the fields that we use in MRI is on the order of 0.1 meters to 1 meter. So first of all, that's in the near to intermediate field regime rather than the far field. But second of all, you'll notice that those wavelengths are way too large to get the sort of millimeter or even centimeter resolution that we might want in imaging. So we need a different way to do it. And of course, that different way, I've already told you, is using magnetic field gradients. So if we have our spin precessing in the magnetic field here, if we put on now a varying magnetic field from one side to the other, the spins precess at different frequencies on different sides. That means that that direction, that spatial direction, is now encoded in the frequency. And this is how, with the Fourier transform, we can get the projection, as you already saw. Now, the gradient is just the spatial derivative of the magnetic field along the appropriate direction. And we can make that gradient point along any direction we choose if we use the right kind of gradient coil structure. And typically, MRI machines have 3 different coils in different cardinal directions so that we can choose freely, which means that in MRI, unlike in CT, for example, we can choose the orientation of our slices freely. Well, okay, so now we have those projections. We can generate tomography in the normal way. And in fact, Paul Lauterbur, in his seminal Nature paper in 1973, envisions just moving those gradients around and collecting different angular projections almost identical to CT, which was just being developed at the same time as well. By the way, I'll just mention that Lauterbur's paper was a 2-page single author paper that founded a new field and netted its author a Nobel Prize. So not too shabby in my estimation. But it turns out there are much better, much faster ways to do this. This we call frequency encoding. But there's something else we call phase encoding, which gets us closer to the interferograms you've seen in radio astronomy. So how do we do that? Well, let's say we put a gradient, a magnetic field gradient of a certain strength on, you can see it here in green, across the body. And let's say we make the strength such that the spins on one side of the body have lapped the others on the other side by precisely 1 cycle by the time you're done with this gradient. Okay, well then you're collecting signal from this whole body integrated against this sinusoidal variation, which means you're getting the first Fourier coefficient. You put on a gradient of twice the strength and 3 times the strength and so on, and you can get Fourier coefficients up as high as you want. Now, I'll just mention that I've just put the sinusoids here, but in fact, of course, these spins are spread out around the unit circle. So what you're getting is this sort of helix, which means we really have a complex modulation, a complex sinusoid modulation, and we need to measure the phase, as I mentioned before. So if we were to add in each new phase-encoded line and accumulate the image, which is not what we do, but is useful for visualization, then you can see, just as you saw in Urvashi's animation, the information in the image is building up spatial frequency by spatial frequency until you get a nice sharp image. And if we now organize all of those lines of data into a nice matrix with frequency encode along one direction and phase encode along the other, we get our K-space, which is the Fourier pair of our image, but it's also the complete analog of the radio astronomy K-space. We just come by it in very different ways, by explicit encoding of Fourier coefficients rather than by interferometry per se. Okay, so let's get to the inverse problem. And here again, you're going to see that same equation that you saw in Urvashi's talk. The forward problem is easy, right? Our signal is just the continuous Fourier transform of our object shown here as rho, with the values of K just being given by our gradient strength. So what do we do for the inverse problem? Well, we do a discrete inverse Fourier transform to get a discrete approximation of our underlying object. Now, if we're clever enough and we have the capacity to organize our K-space in a regular grid, we can just use the lightning fast inverse FFT. So that would be wonderful, and I could end my talk there. But it happens that for all kinds of reasons, we choose our own K-space trajectories. We have complete freedom with the gradients, right? So we have radial and spiral and rosette and propeller and all of these things. Plus, we do something even crazier. We often choose deliberately to undersample our data. And you may say, what in the world are you thinking of here? You have the luxury of full Nyquist sampling. Why would you ever want to throw that away? We radio astronomers have to work so hard to get partial sampling even. I can give you the answer in a single word, it's speed. Because as you saw in that animation, building up those different data points takes time. And that's time that our patients are sitting in the scanner, trying not to move, maybe holding their breath. So if we now deliberately undersample K-space below the Nyquist limit, if we use just a simple Fourier transform, whoops. If we use just a simple Fourier transform, we get all of these artifacts, which you might call our dirty image. So what does the inverse problem look like in that situation? Well, in this case, then, the forward problem looks very much like Urvashi's forward problem. We have all of these different terms, and there are other terms associated with systematic errors in the system. So if your B0 field is not perfectly homogeneous or not perfectly stable in time, you get an additional spatial and temporal variant phase. The detector coils we have actually have different spatial profiles, kind of like the direction-dependent gains you heard about. So that needs to come into the integral. If the gradients aren't perfectly uniform, we get additional phases. The signal decays as soon as we start it off due to different mechanisms. So there's a time-dependent term there. And of course, even our body moves. Plus, on top of that, of course, we have noise, which can vary from coil to coil and in different K-space lines. So rather than trying to address this integral equation, we, again, as you heard from Urvashi, discretize it and frame it in this classic inverse problem format, y equals ax plus n. So once again, we need to solve the inverse problem. How do we do it? Well, when we have non-Cartesian K-space trajectories, we shamelessly steal a technique from you guys in radio astronomy. We use regridding plus an inverse FFT. That's standard, and it was lifted explicitly from astronomy papers. When we can form a relatively simple matrix over here, then we might use matrix inversion, but generally speaking, again, just as you heard from Urvashi's talk, we use regularized optimization. So now that you've seen this, let me go back and talk about reconstructing undersampled data, and I'll show you just 3 different classes of reconstructions that we can use. One is called parallel MRI, and in this, we use the fact that the different coils that we have around the body have different spatial sensitivities. And we use that then to fill in missing spatial information, almost like you do with Earth rotation, for example. That is essentially a matrix inverse and allows us to skip some lines and go faster. About 10 years later, compressed sensing made its way into medical imaging and MRI in particular. And there you can undersample and get a full image, even with just a single detector coil, using an appropriate sparsifying regularizer. And then another 10 years later, we've started using artificial intelligence to now learn regularizers which have better performance, can get us cleaner images and higher undersampling factors, higher accelerations. So now you know how to tell what's where in an image. What's actually the content of the image. And as you've seen, you can take MRI slices in lots of different directions, lots of different angles. But it turns out for even the same structure, like the brain shown here, you can get lots of different image contrasts. MRI is basically the king of intrinsic contrasts among medical imaging modalities. I won't go into the details of how you can get all these different contrasts. Suffice it to say that ingenious investigators over the last several decades have figured out how to pack useful biophysical information into all different parts of our signal, the amplitude, the phase, the frequency, just like you saw from Urvashi's introduction. And so one example of this is functional MRI, which a lot of people have heard about, which encodes information about essentially brain activation into the amplitude of the signal. There's something called diffusion tractography, which produces these beautiful images following the individual nerve fiber tracts in the brain, basically by encoding the diffusion constant of water into the amplitude. And then we have things like this, phase velocity imaging, which into the phase we've thrown the magnitude and orientation of the velocity of blood, say, coursing through the vessels here. Okay, and then just to finish up this part with another parallel to Urvashi's talk, let's talk about the limits of MRI, which someone had asked for explicitly in the first session. And let's start with resolution. So I want to emphasize, we actually don't have a diffraction limit per se in MRI, and now you know why. That's because we use this frequency encoding principle, which gets us way below the nominal diffraction limit for the radio frequencies we're at. And we also have the advantage of near field detectors. So that's why I was particularly intrigued by Ned's slide back in the first session, showing that medical imaging and radio astronomy, MRI and radio astronomy in particular, had very similar D over lambda ratios. That's fascinating because we don't care about D over lambda in the same way without the diffraction limit. So if someone has thoughts on why that is, I'd be delighted to talk about it afterwards. Regardless, though, what limits us in MRI is the number and nature of projections we can gather. And that depends upon the strength and the speed of our gradients. Now, of course, even if we have a perfect image otherwise, if the body moves, it'll mess that up. And likewise, if water diffuses in the course of our image, that will mess things up as well. So those are the limits there. What about signal to noise ratio? Well, you've already heard that the main magnetic field sort of sets the baseline signal and noise. And then also we use these various types of transforms which can affect signal and noise differently. So that can affect SNR. With contrast to noise, really it's a question of how much signal you're willing to burn in order to distinguish two structures. But it's important to remember that despite all of these concrete metrics, in medical imaging, it's the radiologist's eye that ultimately tells you what truth is and ultimately tells you whether there's pathology or not. So clinical information content is really the ultimate arbiter for us. And then finally, for speed, you've already heard that the strength and switching rate of our gradients and also the sensitivity patterns of our coils make a difference, along with our capability to solve the inverse problem. So this comes down once again to technologies and transforms. So in the last few minutes of this talk, I'm going to talk to you about what new technologies and new transforms are coming down the pipeline for medical imaging. And I'll try to organize this by reference to astronomy. But also interestingly, a number of these technologies and transforms are starting to emulate our own evolved biology, which, as I mentioned right up front, is where imaging got started in the first place. Well, obviously in astronomy, when it comes to telescopes, bigger is better. And we have a similar trend in medical imaging. So if you see the early low magnetic field strength scanner prototypes from the first phase here, we've moved up to things like this, the 7T 7 Tesla MR scanner that we have at my institution at NYU. But interestingly, in recent years, there's been a notable trend in the opposite direction. And here I'm showing you a picture of me in a brand new 64 millitesla scanner that we just bought from the company Hyperfine. And this is not without its precedent in medical imaging. In fact, ultrasound used to happen in these big water tanks. And now, of course, we've moved to these handheld transducers. So the question is there a less is more moment coming in medical imaging? One of the problems is that for modalities like MRI, going to lower field means that you're really degrading your image quality. And this is where new transforms can come to the rescue to some extent. AI, in particular, can rescue degraded images. You've already seen that very quickly and schematically using the undersampled data example. But it turns out we can use this for many different kinds of things. And I just want to mention here that rather than doing this type of AI problem blind, you can actually apply some physics in the process too. So one way you can do it is you can unroll that iterative inverse problem that you heard about from Urvashi and include what you know about the physics in a system matrix in, say, a data consistency term, and then just learn the appropriate regularizer. What this means is you're doing a search for solutions that are physically plausible, not just all possible solutions. And that's kind of important if you want not to create a tumor where there isn't one, for example, in medical imaging. And so this is what we call physics-guided learning. Use what you know and learn what you don't. Well, what about this other technological development in radio astronomy of using multiple different detectors ultimately to make a telescope the size of the planet? Is there some analog for that in medical imaging? Well, one thing we can do with help from AI is start to think about coordinating imaging sessions over time. So we can use AI to compare old images to new images to detect significant change. So in a way, this is like giving our scanners a bit of a memory. Now, one other thing I've been thinking about a lot recently is ways we might be able to coordinate among devices, not necessarily different MRI machines, but correlations between expensive images obtained with MRI or other traditional modalities and cheap, abundant sensor signals that we might put inside the scanners. And so this would be in a way like giving imaging a nervous system. If we're lucky, I think there's a chance that these types of sensors and correlations can help us to escape the typical imaging tubes. So overall, what I'm talking about here is really moving from imitating the eye to emulating the brain. And I mean this not just imitating the tasks that humans perform. And in 2016, Jeff Hinton, one of the modern deep learning pioneers, caused a big stir in radiology by saying we should stop training radiologists because machines are going to do their job better than they can in 5 years. It's been 5 years since that prediction, and I don't know a single radiologist who's been replaced. But I actually think that's one of the most boring types of uses of AI for imaging. In fact, as you've already begun to see, AI can allow us to work with different data, undersampled data, different types of data, changing our imaging machines and how we use them. So I hope that you'll agree with me that there are bright new horizons for medical imaging, and that just as radio astronomy is finding new ways to inspect outer space, there are new technologies and transforms that can help us get a cleaner view and a clearer view of inner space. So with that, I'd like to close. First, I'd like to express my gratitude once again to Urvashi, who for me has validated already the organizing principle of this workshop. This seminar series, which is to bring people from our separate and kindred fields together to spark new thinking. I want to thank my longtime colleague and friend, Nicole, for inviting me in the first place to participate. And then I want to say the first thing I did after I accepted Nicole's invitation was to go tell my son, Noah, who has developed an interest in exoplanets of late and is about to launch on a project combining machine learning with astrophysics. And so I vetted my slides with him, and I think he made them much clearer. Any remaining blurriness is entirely my responsibility. And then finally, I'd like to acknowledge my colleagues at NYU who've taught me all kinds of things about both imaging and community. So that's all for me. Thank you very much for your attention, and I'm happy to take a crack at any questions you might have.
Okay, wonderful. Dan, thank you so much for that very clear and informative talk explaining medical imaging to the entire community. So with that, we will start with a couple of questions. On the topic of bigger and better in imaging, in radio astronomy, there's some discussion about whether we need the biggest telescopes for every science case. There are many folks who stress the continued usefulness of smaller, politely mid-scale observatories, because bigger might technically be better, but it isn't always necessary for the science case. Are there any analogous situations in medical imaging?
There are examples all over medical imaging of precisely that. Remember, depending upon what you want to see, you will choose different imaging modalities. So ultrasound, for example, is tiny compared to our big over-engineered MRI machines, but there are many cases where you might want to use that. Likewise, this move that I mentioned towards lower field MRI, the real motivation for it is actually to improve the accessibility of MRI. Because right now, MRI is only done in hospital or outpatient centers that have a pretty high level of infrastructure. So if you could actually bring a cheap, light scanner to the patient, or develop scanners that will work in low to middle income countries that don't have a lot of scanners, there's actually a huge value for vast populations of people. So that's a big old yes.
All right, here's another question. Does MRI or other medical imaging technique have or need a scheme to characterize the instrumental effect, so you can isolate the effect only from the object, cells or tissue, and not from the instrument itself? In radio astronomy imaging, we have such observing schemes called calibration, so we can isolate the instrumental effect. I'm wondering whether medical imaging has a similar issue or need.
Absolutely. I think the theme of this session is going to be, we share a lot of the same issues. So pretty much every medical imaging modality has a whole host of calibrations that are done under the hood that the experts may know about, but the users generally won't. So, when you're actually running an MR scan, if you're sitting inside the scanner and listening to the sounds, often you'll hear this sort of strange kind of tentative clicking and so on that goes on at the beginning. That's frequently a bunch of different calibrations that are being performed. And then there are all kinds of calibrations for the field homogeneity and the gradient linearity and any number of other things. So I showed some of those different terms in the generalized inverse problem. But yeah, we do any number of calibrations using various kinds of detectors to try to get these experimental effects separated out from our images.
All right, yet another question. What does MRI actually image? Is this any hydrogen or only hydrogen and water? And do you get any aliasing problems from weaker responses from other molecules?
Really good question. And I guess in getting all excited about all the intrinsic contrast of MRI, I kind of neglected to mention the key core contrast. Basically, what you're seeing is the density of the MR active nucleus that's operating at the radio frequency you're on, convolved, if you will, by some of the time decay of the signal, which is due to various mechanisms and the local magnetic environment and so on. So what you're seeing at baseline is how much, for example, water, how many protons there are in any region and what their local environment is. Now, that said, there are lots of different MR active nuclei. We don't actually typically get a lot of interference from them, except sort of higher order things, because their frequencies are far enough separated from our Larmor frequency that we choose to sit on that we can filter them out. So there are some effects, but usually when we're looking at a nucleus, we're looking largely at that nucleus. Feel free to jump in and correct me, Nicole, if I ever misstate any of this.
No, that made perfect sense. I was thinking about fat, though. So we do see protons, not just from water. We see protons in other molecules as well, which I know you can probably speak to this more eloquently than I can, but often due to a small frequency shift will appear as another image. We'll have a water image and a fat image, which is a little bit offset from the water, which can sometimes be a challenge, but also can be very useful in some cases if we know to anticipate that in our data collection.
Absolutely. No, that's a great point. And, I already showed you, earlier that we're basically encoding space with frequency. So if the field is slightly different, experienced by some spin, just as Nicole said, it'll show up offset. And so, yes, we have to correct for that, but we also have some of these, some cool techniques to use that and basically get just a water image or just a fat image if we can manage it right.
All right, another question from our audience. Can you elaborate on the kinds of physically motivated constraints you would use to learn your regularization matrix in a machine learning case?
Sure. And this is a longer discussion that maybe we can, I'm happy to carry on beyond this as well. So one of the things we can do, for example, again, because we control our encoding in MRI, I'm going to use MRI as an example, although this kind of thing can work for other modalities as well. We can basically work into our data consistency term a forward problem. We can basically say, we know we acquired data with these k-space values. And now let's compare our working image against something that is, that has those k-space values. So we can basically work in knowledge of the experimental encoding, which means we're never going to choose an image that is not inherently consistent with the acquired data, the data we know we acquired in this pattern. So that's an example of a known thing that in terms of the learned regularizers, we can parameterize that however we want. And one of the cool things is when we did one of these types of networks, we sort of let the network learn a variety of different types of regularizers. And when we looked at what regularizers it learned, one of the first things it did is it learned compressed sensing. It found an L1 regularizer. That was one of the filters that showed up amongst the learned family of regularizers. But it also learned crazy things that weren't even convex, that didn't make any sense to us, but sort of got us thinking about how maybe we could hardwire some of these things too. Bit of a vague answer, but happy to talk more offline.
All right, so I'm going to pose one more question online. And again, just like Dr. Sodickson was saying, we can move any of these conversations to Slack if people still have open questions. Okay, so the question is, in general, how does the medical imaging community view machine learning or AI approaches? Are they open-minded, skeptical, viewed as snake oil, etc.?
Well, remember Jeff Hinton and the proclamation that radiology would be dead in 5 years. What I would say is there's been a dramatic evolution, and it also depends who you are, whether you're a practicing radiologist or you're a medical physicist like Nicole or me. So initially, I think in the field of radiology, there was a lot of fear. Is this going to replace us? What does it mean? What does it mean for our jobs, our livelihoods, our role in productive society? Over time, a much more collaborative picture has developed, and it's encapsulated nicely by a succinct phrase by Kurt Langlotz, a colleague at Stanford, who said, AI won't replace radiologists. Radiologists who use AI will replace those who don't. And then just the last thing I'll say is, for the medical imaging scientists, they jumped on AI very avidly and very quickly and tried it out for all kinds of things, basically from day one. Is there skepticism? Absolutely. Everyone hates sort of the blind learning. When people talk about using generative adversarial networks, GANs, to reconstruct images, many people, myself included, wince, because the whole point of medical imaging is you need the image not to just be pretty, but to be true to that patient. You can't create a nice hallucination of a cancer and then tell the patient that they have it. So there's a lot of nervousness and skepticism, but also a lot of exploration of the capacity for these tools.
Well, I think that is a wonderful way to wrap up. So I would like to once again thank both of our speakers, Dr. Rao, Dr. Sodickson. I think this has been a really wonderful exploration of the similarities and differences of these two fields. And I think this was a really great session.